Thanks again for the opportunity and to present this actually new method that we developed and our package alongside it to detect copy number variants from SNPs. I am representing the Milesi lab from Uppsala University with my co-authors Jay and Pascal. So why is it important to detect copy number variants? Copy number variants are a type of structural variant. When the allele numbers are unbalanced in structural variants, we classify them as copy number variants, which could be insertions, deletions, or duplication. And from studies, we know that this actually affects the gene dosage, because if you have more copies, it actually changes the functionality of that gene, which in turn affects the evolutionary trajectory of a species or a population. And we know that they are involved in early stage divergence of populations of species, especially in local adaptation to environmental conditions. But the problem with detecting copy number variants is that there is no convenient infrastructure for someone like me or someone who doesn't have a good knowledge of all the programming languages to detect this, because there are few methods, but they're very hard to follow and do copy number variants detection with ease. So that's what we wanted to do here. We developed a method, not only a robust statistical method, but also an easy to use R package. So the approach is we took the inspiration from these two papers, especially this McKinney paper in 2017. They basically they described that if you plot the proportion of heterozygotes in using SNPs and their allele ratio from the depth values from a SNP detection, if the genes are not duplicated, they should follow this line, which is 1-to-1 ratio of allele. Whereas for duplicates, it could be from anywhere from 3-to-1, 1-to-3. So this is basically the idea. And Doran et al. In 2020, they used this method and successfully kind of used to detect duplicates or copy number variants in red lobster. But the problem here is that the analytical scheme and the pipeline they have developed is data specific. It's only for red-seq sequencing, and it's hard to use for any other sequencing methods. So that's why we wanted to develop this method, and we split this approach into two. The first one is detecting excess of heterozygotes, where proportion of heterozygotes is always bigger than expected from the Hardy-Weinberg equilibrium in duplicates, as you can see here. So we used this method and we did several simulations to see how this works. And this is from our simulation. So what we test here is that if a SNP is not following this curve, if it is outside of here using Fisher or chi-square test, we can say that it's a duplicate. The next one is the SNPs deviance. We know that allele ratio calculated from depth values is affected by the total depth in that SNP, or the number of samples or number of heterozygotes for which we get depth values. This is not very well addressed in these two studies, especially in this one. As you can see here, they have put an arbitrary threshold to determine the duplicates. So here, what you see in this black cloud is the singletons, and the red ones are duplicates. So they have used an arbitrary method whereas in the McKinney paper, they haven't gone much into this variation. So we did that. We checked that actually with some simulations. As you can see, as the depth value changes from 1 to 200, the expected median allele ratio changes a lot from 1 to 0, whereas it should be 0.5. It's the same with number of samples. It is much more for depth variation than sample variation. So to test this more, we did a simulation of 10,000 simulations, and we plotted that against the median depth coverage and number of samples. As you can see here in this red area, this is low confidence. That means that varies the values you get, the median allele ratio values you get varies from the expected this much, giving us a very low confidence. As you get to this corner of the plot, you can see you have a very good confidence. So we need to account for this in our detection, which is what we did in the next steps. So our approach to this is using a z-score per SNP per sample for heterozygotes, where we use the median or mean coverage of a particular SNP depth value and one of the alleles, and it's divided by the standard deviation for binomial distribution, where P is the probability of occurrence or the success rate of that particular allele. Once we calculate that per SNP per sample for all the heterozygotes, we get the sum of the z-score, and it should follow a normal distribution, which we test again against a normal distribution. And if it falls outside of the normal distribution, we flag it as a duplicate. So now we have excess of heterozygotes, and we also have this z-score, which we use for detecting the SNP deviants. So if we put these together, these two steps together, we can use that to detect duplicates. So this plot shows real data using this method detected for duplicates. And here in the black cloud, as you can see, it follows the natural spread of expected from variation, SNP deviation, and also we can see duplicates from here, and it actually follows this pretty much this curve. We also have added another component to this, where we flag some of the SNPs when the minor allele frequency is too low to actually detect duplicates. So that means we have too few heterozygotes for those SNPs. So we have a couple of examples. I want to reiterate here the most important thing is that, so these three examples show different kinds of sequencing data. For example, the first two are for RAD-seq sequencing data, whereas for PCIABs, we have exome or target capture data. So we can use this method for any type of sequencing, whereas McKinney or Durant methods were used only for RAD-seq data, which was not quite adaptable to other methods. So if I take a comparison of our method and the Durant method, as I explained before, they used an arbitrary threshold to demarcate or separate the expected and observed duplicates from the singletons or single copy SNPs. Whereas in our case, we follow the natural spread of the curve because we take a statistical approach to using our alleles to separate these two. So this means we can use this without really having to take a look at this plot because that's what they did. They plotted, first made all the calculations of the median allele ratios and then plotted it and look at the plots and decided which threshold they had to use to detect the duplicates. Whereas we developed the statistical method, which will automatically detect all the duplicates or separate them from singletons without the user having to check if it is outside or inside the threshold, or even decide a threshold. So we wanted to check if this actually worked, if we really detected a biologically meaningful duplicate. So we made a PCA of singletons, which are non-duplicated SNPs, and using genotypes, and we don't see any structure, population structure there. And then we use the duplicates that we detected, but we use only the genotypes. We see some separation here, some population structure, but when we use the read depth values from the duplicates, you can see clear structure here, separation, populations into different clusters. So we know that this method works. So we used the red lobster data actually for this. There's another thing that we are currently working on. As I said before, here we assume that the chance of one or the other allele occurring is 50-50 or 0.5 as in here for calculation of the standard deviation, which is when we checked the data, which is always not the case. From here, you can see if you plot the reference allele and the alternative allele, you can see some of them actually follow a correlation, which means that there is some sort of bias in the detection of or in capturing the two alleles. This could be due to probe bias or any sequencing errors, especially in the case of target capture. You can see this bias. So we are currently working on using a better p-value for this calculation to further improve the detection. So we have now put all these methods into an R package because we wanted to make it easier for everybody to do a copy number variation detection. So we developed this R package called RCNV. Currently, it's hosted on GitHub. We are planning to put it out there in CRAN pretty soon, as once we have finalized the package and at least the first part of the package and manuscript for it. I will briefly go through how we can use the package. So the package includes the functions starting from importing VCF files, either filtered or non-filtered VCF files, and then detecting the duplicates. So you can, if it is not a filtered VCF file, you can filter for missing samples and SNPs, and you can also test the relatedness and heterozygosity, and then you can generate genotypes or depth tables. From you can actually start here from if you have a filtered VCF, you can already generate a allele depth table. From there, you can get the allele ratio information, and within 3 functions and much faster pace, you can detect the duplication, and you can make a plot. Plot is not the only output. We can output a VCF file of SNPs of singletons, duplicates, and another table with all these duplication status from all the SNPs. So we are further planning to develop this into adding more functions so that we can use VST, which is similar to FST. Instead of genotypes, we use the depth values from populations, and we can use this in downstream analysis, like environmental association analysis. And also, we are also developing another method to method and functions to detect relative copy numbers from these detected duplicates, so that we can use.
Can you hear me?
Yeah.
Okay, sorry. Maybe my microphone wasn't working before because time is over already.
I'm actually done.
Would you quickly want to finalize the slide, and then maybe we can go to the question and answers?
Right. Yeah, I'm actually done. This is my last slide. So yeah, that's pretty much our package, and we will, as I said, this is the package in the development, and we will really appreciate your comments and take on this. Thank you.